everybody. This is Dr. Gilchrist with the promised screencast for brain and behavior. So, I want to take some time to talk a little bit about how neurons actually are capable of communicating with each other. And so, one of the things that we know is that in general, neurons have some pretty interesting electrical properties. So one of the things that we know is that the neuron is covered, like all animal cells are, with a semi-permeable membrane. Now, what this means is that some ions, which are charged particles, can come in or leave the cell, but other types of ions are not. And so what we tend to find is that outside of the cell, we tend to have a pretty high concentration of things like sodium, which is positively charged. On the other hand, inside of the cell, we have a higher concentration of potassium. And so one of the things that we know is that compared to the outside of the cell, the inside of the cell carries a bit of a negative charge. In this particular case, that translates to about negative 70 millivolts. And so one of the things that's interesting is that this neuron generally is not welcoming to sodium. Sodium cannot enter the neuron unless a certain electrical potential, a certain threshold has been reached. Prior to that, sodium can't enter. And so there's a very, very delicate balance that is being held between the inside and the outside of the cell. On the other hand, we have things like the sodium-potassium pump and other ion channels, especially for ions like potassium, that allow them to come and go freely. But for whatever reason, sodium really can't. And that contributes to what we call the resting membrane potential, at negative 70 millivolts. So one of the things that will tend to happen is that action potentials will often be how neurons talk to each other. So there are three particular phases that will happen at this point. So first of all, what we tend to find is that the connections between neurons are numerous. There are 86 million, if not more, different connections, 860 million, if not billion, of connections that are being formed between your billions of neurons, which means that at any given point, a neuron is communicating with possibly hundreds or even thousands of other neurons. And so what we find is that neurons will transmit their message to this particular neuron. This particular neuron, which we're going to call the postsynaptic neuron, will receive that information. And then it will integrate all of the different inputs like a calculator. So some of the messages are inhibitory. They're going to basically inhibit the neuron. Other messages are excitatory, and they're going to potentially contribute to an action potential or a neural impulse. And so that particular neuron is going to add up all of the different inputs that it receives. And if the summed inputs reach a particular threshold, the sodium channels will open and rush into the cell, and the action potential will begin. So here's kind of what this looks like. An action potential goes down the neuron, releases some neurotransmitters. That neuron that is receiving the information will sum those inputs and determine whether or not to reach threshold. If it does reach threshold, we actually find something pretty interesting. So here is our resting state potential, as you can see right here. As long as that threshold is reached, the action potential always happens the exact same way. What's going to happen is the cell is going to rapidly become less negative. We call this a depolarization, and it's going to peak at around positive 40 millivolts. After it peaks, the sodium channels are closed, and the potassium channels are very, very slowly starting to close. And at this point, we start to see the cell become what is called hyperpolarized. It becomes more negative. And actually, for a little bit of time, we have a refractory period where the cell is even more negative than its resting state. And during that refractory period, it's very, very difficult to get that neuron to fire, or it would require stronger stimulation, after which the neuron will return to its resting state. But what's kind of interesting about this is it doesn't really matter how strong your stimulus is. If it's strong enough to get to threshold, you're going to get the exact same action potential. The action potential does not change based on the strength of the stimulus. So it's kind of like flushing a toilet. It doesn't matter how hard you flush that handle. If it's hard enough to make the toilet flush, the toilet will always flush the same way. You can see a few failed initiations. This is where the neuron is stimulated, but it's not strong enough to make it to threshold, after which it returns to its resting state. In this particular case, that's kind of like when you don't flush a toilet hard enough and it looks like it's starting to flush, but then it just stalls. So action potentials travel down the axon. So this change in voltage from negative to positive back to negative continues to travel down the axon, as we saw here, to the terminal buttons. And you have to have an action potential for these neurotransmitters to be released. So the neurotransmitters are going to be sent in that little gap between neurons. This is what is sometimes referred to as the synaptic gap or the synapse. And that might be a quiz question, just saying. So these are going to send messages to postsynaptic neurons. Now, the message that is sent depends on the type of receptor that that particular neuron has. Interestingly enough, there is research on different types of drugs that actually help us understand a lot about how neurotransmitters work. And as you're about to discover, drugs are either going to be what is called an agonist or they're going to be called an antagonist of the neurotransmitters. So here's how they work. Let's go back to that image that I showed you before. And in this particular case, we're going to magnify this particular area right here. This is an instance where a neurotransmitter has been sent into the synaptic gap and it's making contact with what is called a receptor or a receptor site. And so what you're going to notice is that this particular neurotransmitter fits the receptor site perfectly. So you have very specific types of receptors all over your neurons' dendrites so that different neurotransmitters can make contact with that cell. And they're very, very specific. So dopamine receptors are going to look differently from serotonin receptors. It's designed to fit a particular neurotransmitter. Drugs work by basically hijacking how this receptor site works. So in this particular case, here's our neurotransmitter. On the other hand, if you look right here, this is what is referred to as an agonist. An agonist is designed to mimic or enhance the effect of a neurotransmitter. In this case, it's partially because it is similar enough in structure to the receptor site. So in this particular case, morphine, as you see in this image, can actually mimic and enhance the effect of our endogenous opioids or endorphins. They operate similarly. On the other hand, we have what is shown here. This is a particular case called an antagonist. As you can see, just like we saw with the agonist, it has a similar structure, but the antagonist is designed to inhibit the effects of the neurotransmitter. It basically is preventing any of the neurotransmitter from getting to this receptor site by effectively blocking it. So one such example that we might see is a case like curare. Curare is a type of poison that can be used to basically act as a paralytic. It produces a flaccid paralysis. That means that the muscles can't contract, they just go limp. And so it does this by blocking receptors for a particular neurotransmitter called acetylcholine. Now let's talk a little bit, I'm trying to move that. Let's talk a little bit about what these neurotransmitters do because they are involved in so much of what we do. So here are a couple of particular ones. What you're looking at here is an effect of acetylcholine. Acetylcholine is really critical for motor control. Anytime that you contract a muscle or use a muscle, you can thank acetylcholine for being able to move your muscles. It is also critical for attention and learning. Now, if you look over here in this image, this is a woman that's being injected with Botox. Botox is basically a drug that is derived from a bacterium toxin called botulism or botulinum toxin. When this is applied in a drug form like Botox is, it is an antagonist to acetylcholine, and like I mentioned with curare, causes a flaccid paralysis. So you inject Botox into the muscles of the face so that they can't contract, reducing the appearance of wrinkles and fine lines due to movement of the face. We have neurotransmitters like epinephrine and norepinephrine, which are really critical for regulating our fight or flight response. They tend to spike when we're stressed or scared or we're being challenged by something. Serotonin is really critical for our mood. Dopamine is really critical for things like motivation, reward. So anytime that we do something that we enjoy or find particularly pleasurable, we're going to get a rush of dopamine from it. It's also critical for things like motor control. We have endorphins, those endogenous opioids. They are also very rewarding and they're also critical for pain reduction. Now, interestingly enough, endorphins are not considered a classic neurotransmitter per se. The others are, in the case of acetylcholine, it's a very simple chemical structure. Epinephrine, norepinephrine, serotonin, and dopamine are all cases of what are called monoamines. They have an amine group. Endorphins are technically neuropeptides, and they typically have not been considered a classic neurotransmitter per se. So I'm going to go ahead and stop here with this before I move on to another screencast about the brain and its many different functions.